This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 446, recorded on June 16th, 2017. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses, coming to you today from a cloudy New York City, 20 degrees Celsius. And joining me here is a tanned Dickson Despommier.
And relaxed, too, Vincent. Thank you for adding that.
Relaxed.
Yeah, no, I had a great 3-week vacation, so I'm excited to be back.
Aren't you always on vacation?
No, come on. I'm retired, but I'm not always on vacation.
Well, hearing what Rich Condit just did on his retirement sounds like a vacation.
No, but I actually, on my vacation, I gave 2 talks, one in Milan and one in Peter Maritzburg.
Cool.
So I had a good time.
Welcome back.
Thank you so much.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. How you doing?
The guy who's on vacation.
Yeah, I'm on vacation. I'm not gonna make up any stories here. I'm on vacation.
Well, Dickson comes here, where he has an office. And he does stuff.
I do.
And so
I love coming here.
Yes, it's working, right? But you just don't get paid.
That's right. I don't get paid.
I need to tell you that it's 93 degrees Fahrenheit here, which is 34 centigrade.
And the humidity?
The humidity is, I'm glad you asked, it is 49%, which is not too bad.
No,
I think in terms of dew point, it's 71 degrees is the dew point,
nice.
Which is, a little humid, but not bad. If you were coming from San Diego or LA, you would think it was humid, but
right.
The heat index is 101.
Okay. So, Rich, on my trip, I had to stop off in Dubai.
What did you do in Bali?
I just changed planes, but it was 95 degrees and 90%.
It was at 6 a.m., right?
Yeah, exactly right. Yeah. It goes up above 42 degrees sometimes centigrade.
The non-permissive temperature.
That's crazy.
Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here today, it's partly cloudy, 85 degrees. I think I'd rather call it partly sunny. Looks nice.
That's right.
Yeah, we don't have any sun here, do we, Dickson?
We're not, and we're expecting rain over the next 3 or 4 days, too. Yeah, I'm okay with that.
Joining us from Western Massachusetts, Alan Dove.
Good to be here. Yeah. It is fully cloudy.
Fully cloudy. Yes.
There's no ambiguity about the conditions here, and it is just probably about to rain. It has that feeling of
that's right.
Rain on the way.
You got it.
Are the leaves on the trees turning upside down?
Occasionally, and we're getting puffs of wind in different directions, and we had a few little sprinkles, but I think tonight we're supposed to get thunderstorms.
That's it.
Wait a minute. Leaves on the trees turning upside down.
Yeah, they turn silver.
Right. So the idea is that they tend to grow in the direction of the prevailing wind, and so when they're showing their backs, it's because the wind is blowing differently from the prevailing wind, which is a possible indicator that the weather's going to shift.
The things you learn on TWiV.
There you go.
See, weather is important.
We're back together after a few weeks of fragmentation. We shall be back together for this, the next, and the next, at least, I think. That's as far ahead as I can go.
Absolutely.
And we'll be in Madison, hopefully.
We will be.
If it all works out.
We will. Should.
And after that, we'll be tired of each other again. I want to tell you about the ASM grant writing online course. Learn the best practices in grant writing. You can participate to receive an overview of the funding canyon, learn what makes grants successful, and receive personalized feedback on your grant. And it's a 7-part online series taking part in the fall of this year. You can register online. The deadline's July 15th, about a month from now. You can learn more at bit.ly/ASMGWOC17. That's 2 digits, 17, all one word. bit.ly/ASMGWOC17.
Just rolls off your tongue.
What's happening with ASV, Kathy?
Well, as we know, it's going to be starting a week from tomorrow. For us, we're recording on Friday, so Saturday, June 24th. For the people who are up to date on TWiV and listening on Sunday, the app is going to be locked supposedly as of midnight tonight. So if you want to go in and really plan your schedule to the minute, then all the scheduling should be the same. If we have any withdrawals after this, we're just going to have to leave gaps in the workshop. If for some reason you didn't get the email about getting your app, then you should email asv@asv.org, and they will forward you that email. Or you can also contact me, and I will forward it to you. It has the information about how to download the app, and also the ever since May 15th out-of-date old-fashioned PDF. That's all I wanted to say.
And when you say the app will be locked, what you mean is that the schedule will be set.
That's what I meant. Yeah, frozen.
Not that you can't download it after that.
Right.
When you said that initially, I thought, wait, the app is going to be. Why did I download this thing?
No. I said it wrong. Yeah,
the schedule.
Yeah.
The schedule.
The schedule is locked.
It'll be etched in stone.
Right.
We have a bit of follow-up, which we collect since the last 2 episodes. We didn't do any. Did anyone enjoy the astronaut, by the way? I've got no feedback.
Oh, yes.
No feedback on the astronaut.
Awesome. Loved that.
Yes.
She was great.
Yeah.
Mm-hmm.
All right. Junio writes, this is funny because Junio used to write us often. It's a Japanese fellow who works at Google, and he stopped, and I'm thinking one day, why doesn't he write anymore? And then this email came in.
Wait, if he's Japanese, is it Junio or Junio?
Junio, I guess, right? I don't know. Is it Junio or Junio?
It's Junio.
Junio?
It's Junio.
Yeah, Junio.
It's J-U-N-I-O.
Not Japanese.
J-U-N-I-O. J-U-N-I-O. Well, I see Junio, and I think Junio.
Yeah.
Well, right.
Junio.
Thinking in Spanish.
And here in New York City, we do that.
Yes.
So every sign is in 2 languages.
This is true.
Why is that? Because a lot of Spanish
We have a lot of
people.
Hispanics.
Anyway,
That's right.
Junio writes coccinelle in Japanese. These colorful round small beetles are collectively called tentomushi. Dickson. Tentomushi. I'm bowing
that
as
sounds
I,
Italian.
as I say that. I know. I tend to give an Italian accent. In Japanese, mushi is a generic term for insects.
Right.
Tentu is the sun god. You can call the sun Otentu-sama. Sama is part of Gochiso-sama. Wikipedia says they fly towards the sun, hence that name. The most common ones are called Nanahoshi Tentu, 7-star sun god.
My goodness.
You can call me that, Dickson. Tentu,
Really?
Nanahoshi Tentu, Sama. There is less commonly used alternative spelling of the name of the bug, which is Beni Musume. It literally means red girl. So in that sense, in Japanese, they are also deemed female, just like ladybug implies female, but not many people know that spelling.
Right.
Lovely. There's a wonderful anime about Mushishi. That's it. It's a little different. Mushishi. The series follows Ginkgo, a man who dedicates himself to keeping people protected from supernatural creatures called Mushi, which are really, as we heard here.
During your discussion of the Japanese beetle,
Insects.
did the subject of roses come up by any chance?
We didn't talk about Japanese beetles. We talked about ladybugs in that show.
Oh, because I've been misled by this.
This discussion is about ladybugs,
Okay.
but it's about the Japanese term for ladybug.
We wondered what it was
Gotcha,
in Japanese.
gotcha.
I've forgotten why we wondered.
I was going to say, I've forgotten.
Well, the reason is because in most of the other languages, lady is part of the name when you translate it, and it wasn't when I got, or I didn't think it was because of the interaction I had with somebody Japanese who I showed the beetle to, and I said the little poem, and then she said, "How do you know it's a female?"
Right.
Because the name had lady beetle in it.
So on another take on that, then, ladybugs, lady beetles are very important for biological control measures indoors to keep aphid populations down.
Yes.
Because their larvae eat them like crazy.
Yeah, we just bought a whole bag of ladybugs to clean up our bougainvillea
Nice.
outside. Yeah.
So there.
Do you know what Gochiso-sama means?
No idea.
After you have a meal, you say that's like a great dinner. You say to the chef, Gochiso-sama.
Okay.
And that's funny because sama must be something common. There are a lot of those common things. Kathy, can you take the next one, please?
Sure, and this is from Akira Ono, who wrote to me in response to my question. Japanese lady beetles. Google translation is helplessly incompetent in this. It is called Tentu Mushi. Tentu means the sun god, or the path of the sun, and Mushi means bug. So sun bug or sun god bug would be the better translation. These insects are thought to fly toward the sun, hence the name.
Okay.
Akira.
There you go.
Alan.
This is regarding 444. Anthony writes, I found interesting the need for an inverter. How many devices are not available for 12 volts? In addition to space, machines that require 110 will be difficult to use in a remote location on Earth. It was good to learn that much of a standard lab can be used immediately in space, perhaps with just minor adjustments in technique. Even so, can better tools be designed? Our comments on experience and limitations encountered being collected and published for manufacturers to review and incorporate in future designs for space applications. Thank you on a particularly great TWiV episode. Even though I basically stay put on a side street in Jersey City, through Twix I learn about topics in a range of the biological sciences, hear Nobel laureates, and now find out about living in space from someone who was there.
That's the idea.
Yes, Anthony continues, let's imagine BSL 10 as a facility in deep space where nothing but data ever leaves. I think this was in one of the Alien movies, wasn't it? What would the space station then be? BSL 7?
Yeah.
Alan, in space, no one can hear you scream.
This is exactly right. Did the astronaut discuss the fact that they're printing their own tools now with 3D printers?
No, we didn't discuss that.
They shipped one up, and they now can manufacture their own tools.
You might have mentioned that.
I think she did.
There was
mention
some
that
discussion.
She 3D printed a centrifuge rotors.
Yes, exactly.
That they shot up, and for the motor it was printed so it could sit on a drill.
It goes into a drill chuck.
Absolutely.
The cool part about, and in fact, after she did TWiV, she went and did the keynote, which was really an interview with Ed Young, and she showed a movie. And the cool thing, she says, when she's doing a manipulation where she doesn't have enough hands for she just puts something there and it stays there.
Exactly.
Hanging in the air.
Exactly.
Oh, it's so cool. But Dickson, the cool thing is you can take a 50 ml conical and pipette fluid. As long as you go in the bottom, the surface tension keeps it inside.
Right.
It doesn't come out. I always thought it would float all over, and they realized, as Anthony says, they're learning these things. I think she is keeping track of all of it, and she's going to write lab manuals for future astronauts to go up and use
NASA is extremely good about recording everything.
Yeah.
I once was in the Singularity University once, and that's where they do the research and development for NASA, and they showed me how they're developing the newest toilets, because that's their biggest problem. Yeah. And they said the best toilets in the whole world were made by the Russians.
Oh, yeah?
Yeah.
And are those up in the space station?
Because they trap all the... No, I won't even go there, but remember, Caleb is listening.
Caleb is listening.
That's right, but that's their biggest single problem. And then it's the odors and the feces.
So it smells badly?
It does.
In space, no one can hear you pee.
Or anything else for that matter.
She actually said that the smell of outer space is... Rich, what did she say?
Mildly metallic.
So basically, when you open a airlock and then you get the air and immediately you go in after that, it's mildly metallic.
How interesting.
But you should go out and try it yourself.
I would love to.
All right, we have a snippet for you today. I ran into Ann Palmenberg last week in Kansas City. She told me about this. It is online as a, accepted, posted online. A Journal of Virology, the JVI accepts, as they're called. It is called Gender Parity Trends for Invited Speakers at 4 Prominent Virology Conference Series. Robert Kalita and Ann Palmenberg from University of Wisconsin-Madison.
And this is open access.
As it should be. Everyone needs to look at this. So it's the original PDF. It will be eventually set into lovely type. But you can still read it because the words mean the same.
They do.
Right?
The formatting is a little annoying, but the words are the same. Yes.
So here I'm going to briefly summarize this since it is a snippet. There is a perceived underrepresentation of female speakers at big meetings, science meetings, and they say, what are the data? We don't want to just say this and not have the data. So they collected the rosters of invited speakers over the last 35 years for 4 international virology conferences: American Society for Virology, the International Herpes Virus Workshop, the Positive Strand RNA Virus Symposium, and the Gordon Research Conference on Viruses and Cells. And they looked at all these data, cross-indexed them, and in fact, they are available online as spreadsheets. And what they found was all 4 conferences show a clear proclivity for male-dominated invited speaker lists. There is a shift toward parity with all these meetings, but at different rates. Mm-hmm. And selection of a larger percentage of first-time participants and the presence of a woman on the speaker selection committee correlates with improved parity, parity being 50-50, of course. Mm-hmm. And they also collected session chair information for the herpes workshop and the GRC, and they display a strong male dominance over time, that is session chairs, that is also eroding.
Now, parity is scored as 50-50 in this paper, but it is important to note that if you wanted actual representation in proportion to the population as a whole, you would actually be aiming for something like 55% women.
That's right. So that makes the question of what is the composition of the membership of those societies in terms of males and females?
They address that in the paper,
yes.
Not necessarily the societies, but the meeting attendees or the principal investigator / faculty / senior scientist or whatever scientist roles you would draw your speaker list from. Mm-hmm. And that information is hard to know. But they do address the populations, at least of attendees for 2012 and 2017. 2012 and 5 years before that, 2007 ASV at Madison. And I think for one other meeting they had that information.
2006,
Yeah, and I didn't mark that.
So 2000.
Yeah, I think they
60-40.
Yeah, in the discussion they talk about ranges from 40-60 to 45-55 female to male. So women are underrepresented to some degree in the population of attendees at the meeting.
Yep.
But in any case, they are far more underrepresented among the speakers.
They also note that they couldn't figure out first names all the time.
Right.
Right.
And it would be important to see how many people get grants and what the distribution is there too, because those are the people that are...
whole other can of worms.
Yeah, but they're going to give papers though, right? And they'll go to the meetings, and so therefore that's the whole thing.
So let me just briefly tick off a few stats for ASV. Anyone else wants to add others, you can. 82 to 2017 for ASV, 77% of invited plenary keynote talks, these are the big ones in the morning that are invited by the president. They're delivered by men, 77% men, 23% women.
This is for 2017?
The whole year, 82 to 2017. That's the overall.
The whole history.
The whole history.
All history, okay.
The best will happen next week, okay? But next week hasn't happened. It could change. You never know. But if it happens as it's planned, the highest will be next week, which will be 53%. And I will tell you that
for the keynote presentation,
for the plenaries, plenaries, not keynote.
Excellent.
Plenaries. Now, until next week, the highest percent female in ASV history is 2015 at 52%.
Wow.
Who was responsible for that conference, Vincent?
I don't remember. Does anyone know?
Oh, yeah. So say, what was your percentage again, Vincent?
52%.
52.
And the highest ever for a male president, okay?
Right.
Yeah.
And I wrote Ann Palmenberg, and she said, you're damn right, and you should be proud of it. You also get a prize for the fewest total repeat speakers. So that's another important metric, because if you pick new ones, you're more likely to pick women instead of picking, as she says, dipping into the old guard well. She was president in 2008. Her percentage was 47, and the highest by 17% in the previous 27 years. Mm-hmm. But after that, once the spreadsheets were available, the tide really seemed to turn, and you were a big part of that. Transparency has an effect. And she says, this is also interesting. All the years I was in ASV trying to affect change, who knew that all one needed to do was make a spreadsheet?
Right. And so the highest percentage of female speakers prior, or the first really high one was when Roz Sandry-Golden was president in 2010, and we all noticed that. I was one of those invited speakers for a plenary session. And Roz was also the first one, or she's the one who had the highest number of state-of-the-art speakers that were women the year that she was, one of the 2 years that she did that as program chair. Looks like 2008 for that.
I must say that one year it was 0% women.
Yeah.
Can you imagine?
2 years for ASV. Early years, 1982 and 82.
It's hard to imagine. In fact, it's the way life used to be.
Yeah, the worst 2 years were 82 and 87 at 0%.
Yeah.
And I would argue that among you, Roz, Becky next week, and you, that probably there's not statistical difference between 50, 52, and 53.
Yeah, they're all good. I mean, I think you need to maintain this to encourage women to go in the field, because they say, look, a lot of women speakers, I can do this. That's possibly one of the more important reasons to do it, right?
Yeah.
Well, and also speaking at conferences is a major route to advancement in the field.
Yep.
That's true.
Right.
So if you want women to stay in the field, then they need to be able to advance in the field, and they need an even shot at being able to have the opportunities. And so this is all part of that whole thing.
There you go.
It's also...
Sorry, go ahead, Kathy.
It's also not stated in the article, but one of the first things I thought of is how important it is as a role model effect. Mm-hmm. And I tried to find other literature about this. The only thing I can think of is a study about students at the Air Force Academy, and among the top women students, if they had a female professor for their science classes, they were more likely to let's see, I've got it here in the abstract. The likelihood of taking future math and science courses, their likelihood of graduating with a math, science, or engineering degree was much greater. And so that's not exactly the same thing as the role model effect. And in fact, there at the Air Force Academy, it didn't happen for, there was no gender difference when you had professors teaching humanities and social sciences. So this was particularly noted in math and science, that having those role models as women helped the top female students stay in science and engineering. So here, too, if you have women giving talks and convening sessions, that was particularly something that they looked up and discussed in this paper that we're talking about, as the conference, the session conveners. And when you have more of those, it's likely to be better and give more opportunities, as Alan was saying, that for advancement for careers.
I understand there's going to be a session on this or a talk on this at ASV. Is that right?
That would be news to me.
Because Anne, in Anne's email, she said this topic will be discussed at ASV. I didn't know what she meant by that. It sounded like there was some kind of formal thing. But
I don't know either if it's... Yeah.
Okay. Do you... what was I going to ask you? Do you agree? I forgot. All right, maybe it'll come back. They conclude our historical record is not one to be proud of.
That's true.
And they say, just by bringing this to the light, just by saying, look, this is what we've been doing, transparency, that helps because people go, whoa, I didn't realize that. Well, but they should have, right? And so they give a couple of suggestions for how to get around this. Yeah. Yes, go ahead.
Oh, yeah, the 5 numbered points of how to fix that. And they also acknowledge that this kind of study has been done in the ecology evolution field, and Arturo Casadoval did it for ASM as well. And I would really like us to do it for the virus specialty international meeting that I attend, which last year was really, really bad, like the early 80s. The specialty virus I work on. I was trying to avoid.
So you have your own meeting.
Yes.
Okay.
It was particularly bad on speakers and session conveners being mostly male. So.
And I think in the article or in Anne's email to me, she said the HIV meetings are thinking about doing this as well.
Mm-hmm. Yeah, you just need to do a spreadsheet, but probably you don't even need to do the spreadsheet. You just look at a couple programs and you can figure it out.
So
but it's nice to collate the data because you're dealing with scientists and you can point to this and say, hey, look, here are the numbers.
Yeah.
And they speak for themselves.
Yeah.
Anyway, it's a good article. It's open access. Everyone should have a look at it. And
yes.
It does, and kind of positively, it says the American Society for Virology state-of-the-art program is a shining example of an outstanding balanced program featuring primarily first-time speakers who deliver compelling presentations. The goals we should wish to achieve are not out of our reach.
So there is a series where you try to you have to get different people each time, right?
There have been repeat state-of-the-art speakers. Mm-hmm. Those statistics are in the paper, but in the last several years, I don't think we've had repeats.
Yeah, because one of their arguments is if you don't pick from the well over and over, you're more likely to have a better balanced speaking panel.
Mm-hmm. They have a cool Venn diagram in here on how many speakers have attended more than one of these meetings.
Right.
That is the herpes workshop and ASV, or ASV and the Gordon Conference. And out of hundreds we're talking about, there are 5 who have spoken at all 4 conferences. And I really want to know who those 5 are.
Yeah, how does it talk? Positive strand
Actually,
RNA.
I was thinking I wouldn't be at all surprised if one of them was Joan Stites.
Could be.
Yeah, and I think maybe Anne was, because I think she went to the International Herpes Virus Workshop to speak there. So, but yeah, how do you speak at the positive strand RNA virus meeting and the herpes workshop?
Exactly.
Well, sometimes meetings invite people from other viruses just to hear what's going on.
I was thinking about this.
You're not obliged to just work on one virus. You could work on several.
That too.
Right, yeah.
I mean, you've done that,
it would be a
Vincent.
structure person.
Or
an immunity person or something like that.
Right.
Well, I guess we can go to the spreadsheets and figure it out.
Yeah, spreadsheets are great.
Right.
Check it out. And I thank Rob Kalita and Ann Palmenbrook for doing that. And Ann said, Rob has never been mentioned on TWiV before, so he was.
I thought we did one of his papers.
I thought we did. I looked it up and I found his name in a letter from TWiV 192. I thought we did a paper though. I really do. But Rob, if you would like to be on, we can do that. Just tell us which paper you want us to do.
Yeah,
you're the one to tell us. Now, Kathy, you had some other interesting thing to discuss.
Yeah. So I was just back from study section, and that's another place where you get to meet new people. And so, Vincent, I don't know how long ago it was, but you picked this, I think you picked as your pick of the week, the Grant Support Index thing, which I nicknamed the point system, where there was a proposal to limit the amount of grant money going based on points for certain types of grants. And while this met with some happiness in some camps, it was thought that maybe it wasn't such a good idea. People would game the system, and then they changed it a little bit, and that probably didn't really make it any better, maybe made it worse. And I think they've shelved that, from what they say, they've shelved that. And they have this new thing called the Next Generation Researchers Initiative, or NGRI. And so that was talked about in the NIH director's comment, and there's a whole information page about it. The gist of it is that they want to try and increase the number of NIH-funded early-stage and mid-career investigators. And so they're going to extend the pay line for R01 applications or their equivalent from early-stage investigators, hopefully funding everything down to the 25th percentile or something receiving a score of 35 if they're not percentiled. And so they're also going to provide additional support for mid-career investigators defined as those who are within 10 years of receiving their first NIH R01. What that doesn't include, which was pointed out to me by one of the people at the study section, is that there's this other category of people, the new investigators. So early stage investigators are defined as someone who is within 10 years of completing their terminal degree, in other words, their PhD, or within 10 years of completing their medical residency. And that category then will disadvantage somebody who, for instance, might have done 2 postdocs or might have taken time off for maternity leave or things like that. And so she was saying, it would be great if we could write in like to the Open Mic blog and comment on this, or to the public input at od.nih.gov link to say that we think that maybe this new investigator category, which does include her, should be looked at as well. So a new investigator is someone who has not previously competed successfully for a substantial NIH research award, which basically means for an R01 or equivalent. If you've had an R21, you can still be considered a new investigator. And I just ticked up the number of new investigators versus early stage investigators that we had. So the early stage investigators grants that we reviewed, that in other words, the people within 10 years of their degree or completing their medical residency, was 4, and we had 13 new investigators out of a total of, I can't remember, maybe about 60. So there's like 4 times as many people, 3 to 4 times as many people in this new investigator category that doesn't seem like it's going to be picked up in this NGRI. So that's something that maybe we can get them to fix.
Yep. I have a staff scientist in my lab who's passed 10 years from her PhD, and her grants, she sent in a bunch, they don't get funded. And one of the common criticisms, she has never had an R01. Well, that is kind of a silly criticism, really.
It's a catch-22. It's a catch-22.
So give her one letter.
Yeah, and for an R21, you shouldn't have to have had an R01.
That's mostly what she's applied for. And for that, I mean, it's a little silly. Anyway, I think this group has to be accounted for in some way. And I don't know. I kind of like the point system. So enough people made noise and they decided to can it.
So I've been away for 3 weeks, and during that time, I heard nothing but anti-Trump comments wherever I went. And the biggest one I heard from my email, of course, was the budgets. And the budget's actually reversed over what he was going to do. Have we discussed that in my absence, and I'm just coming in late?
The NIH budget got a $2
big
billion from Congress because they said no.
Because they want to. Because
what's going to happen next year, we don't know.
See, Trump can only go 8 years at max. These guys can go on forever in Congress, so they don't want to get diselected just because they've dissed some important stuff. So it's,
You mean like healthcare?
Yeah, that. Oh. Yeah, that. So, I don't know.
Anyway, people can write, there's an email where people can write in and give feedback, right?
Mm-hmm.
Yeah.
Yeah, and I looked in the first comment on the Open Mic blog when I looked about a half hour ago was somebody saying that this disadvantages the new investigators.
Yeah. But we had an award when I was coming up through the ranks called the Career Development Award, which I got, fortunately. So that paid my salary.
Did it develop your career?
Yeah, it did, actually. I had a research grant, and I had a Career Development Award. So that frees me from university activities for 5 years. I can go ahead and develop my science, and that was a big advantage for me.
Well, but at that time, the amount of funding relative to the number of researchers was vastly different.
No
the
question.
Current landscape is just a different world.
I'm not saying you'll do it again, but I'm just saying that I had the luck and privilege of taking advantage
the fundamental problem here is that there's too little money being distributed to too many people and done in a way that kind of in a mirroring of our overall economy concentrates an enormous amount of wealth in a small number of hands.
That's right.
And so
that's the American way, Alan. Sorry.
Well, only since the Reagan administration, but so the original proposal to simply cap the number of R01s or other major grants that a lab could receive was a simple and direct approach. But of course, the labs that have multiple R01s are exactly the same people who control the levers of power, and they screamed when this came out, and
true.
So it's not getting done that way. And so now they're scrambling to find some other way to plug the same gap while still enriching the people at the top of the heap, and I don't think any of these solutions are really going to work out.
Limiting the number of grants to 3 doesn't solve the problem as long as you keep asking for more money for those 3.
It doesn't solve the problem, but it does redistribute the money to further down on the pyramid. And somebody who's got 4 R01s in their lab is not going to be out of a job if they don't get their fifth one renewed.
No, that's true. But
whereas somebody who has 0 is not going to stay in science.
I hear what you're saying. But there should be a cap to the amount of money per grant. And if you did it that way, it would be different.
Or a cap to the total funding per lab.
Yeah, that's true.
I think if you phased that in over a period of a few years...
There go the empires.
There go the empires, yes. But if you phased it in over a period of a few years, it would not have to be terribly disruptive. The problem, as I say, is that the people who wield outsized influence are exactly the people who would lose in a system like that.
But of course. I mean, they end up heading up institutes, basically, right?
They do, yes.
Well, my pick relates to this. So stay tuned.
Okay.
Here's an arc. We have a paper for you. It's called The Genesis and Pathogenesis of 1918 Pandemic H1N1 Influenza A Virus, published in PNAS, and it's by Michael Worlby, Guanju Han, and Andrew Rambeau from the University of Arizona, University of Edinburgh, and the National Institutes of Health. And this is all about the 1918 influenza pandemic virus, which killed about 50 million people. So I think this paper is interesting because we didn't have that virus at the time. We didn't isolate any influenza virus until 1930. Now, this particular virus was recovered from archival material many years later in the 2000s. But there are a lot of interesting questions about this virus. Where did it come from? Why did it kill so many people? And it has a very unusual mortality curve associated with it. So a lot of interesting questions, and I think this article addresses it. So that's why we're doing it. It's a 2014 paper, but I happened to hear Worlby talk about this a couple of months ago, and it piqued my interest. He talked about this, so I said, someday we will do it. And so there is the lag time for TWiV, a couple of months paper. All right. So here's the questions that they address. Where did the 1918 virus originate? Why the weird mortality pattern? Now, typically flu kills the very young and the very old, but in 1918, the adults 20 to 40 years were also very susceptible. But the very old were okay.
That's right.
You know, they had less mortality than previous flu outbreaks. We have records of all this. A couple of other interesting things that maybe everyone doesn't recognize. The virus infection was clinically unremarkable in over 95% of patients. So although we hear it killed 50 million people, a lot of people got infected and were okay. And most of those fatalities were a consequence of secondary bacterial pneumonia, because, of course, antibiotic use was not widespread at the time. Now, interestingly, after this pandemic was over, the mortality patterns went back to the very young and very old being the most likely to die from inspection.
So not only do we not know where it came from, we don't know where it went.
That's right. And that's what they address here. Very interesting paper. And what they use here, they take a lot of data. They take a lot of sequence data. They take a lot of seroarchaeology, that is, serum samples that have been stored from people who lived then and have lived since, and we have antibody titers to the different influenza virus strains. And they put this all together. And a big part of this is a host-specific molecular clock. It's not battery-powered. It is a host-specific molecular clock. Now, a molecular clock is a software algorithm that uses mutation rate to figure when two viruses diverged, essentially.
Or two of anything.
Two of anything. Since we love viruses here on TWiV.
Yes, we're going to talk about
You could use it for anything.
Yeah, they do that for mitochondrial DNA, right?
Yeah, exactly.
Sure.
You can use it for clams if you'd like.
For clams.
Yeah, or anything that has something to compare. It could be nucleic acid or protein, for example. And so they, in this paper, they use a host-specific rate, which is unusual, and they say that you need to do this because it evolves differently in different hosts, and it's the only way you can infer how the virus went from one host to another, the timing of that and its directionality. So this is what is different about this approach. You say it gives us a real indication of what viruses went where. So as I said, they combine this with epidemiology, seroarchaeology, evolutionary, and they come to interesting conclusions. And the main conclusion, here it is: childhood exposure to different hemagglutinin and neuraminidase subtypes is a key factor in explaining these age-specific fatality or mortality patterns that happened in 1918 and other times as well, as we'll see. So your first infection is really important, and that can determine how you react to flu for the rest of your life. So you can imagine this has huge implications for vaccines, right?
Yes.
Right?
No question.
So a lot of kids now get their first flu vaccine. That's their first encounter with flu, and that's going to influence their response forever. And so if a new virus emerges, they're not going to be very good at responding to that. So anyway, we'll get to that.
I just wanted to inject something. The fear that this pandemic put into people was so intense that following the end of the First World War, many American troops refused to go home.
Really?
And imagine what it must have been like over there in the trenches, right? And they refused to go home because they knew if they did, they're going to die from flu. And a lot of them went home, and a lot of them died.
And they were in the right age group, right?
Yep, absolutely.
At 20 to 40.
Of course, a lot of them died of flu in the trenches as well.
That's true. But they didn't want to go home after the war. Imagine you're homesick. You want to go home. You're missing all of the things that you're fighting for, and then the next thing you know, you're reading that your aunt just died and your uncle just died and your brother just died because they were still home. Now you don't want to go back.
All right, here's what they found.
Wow.
I'm going to tell you what they found because we don't want to go through the individual data in the analysis. What they found first, their clock analysis, their host-specific clock analysis shows that the H1 from the pandemic, remember 1918 was an H1N1 influenza virus. It emerged from an avian source around 1901 to 1907.
And this is why a few years ago when we had an H1N1 strain, people bugged out. One reason.
Second, at that time, for the first time, as far as we can tell, influenza virus went into pigs. Didn't infect pigs before then. And that virus that went into pig came from the human lineage at the same time.
They knew this because the pigs were dying from this?
Yeah, they started to die. There were outbreaks of swine influenza, whereas they hadn't seen that before. I think these are pigs raised for consumption, right? Mm-hmm. As far as wild pigs go, that's a different story.
Right.
All right. After the pandemic, a seasonal H1 virus circulated for many, many years, up until 1947, I think it was, or 50, maybe when an H2 emerged. That H1 did not emerge from the pandemic H1. It was from a separate virus that was circulating. It was a different lineage that had been circulating around 1907.
Different birds.
Actually, according to this in Figure 2, their phylogenetic tree, it looks to me like that H1N1 is circulating up through the 70s.
It's still around, but there was an interruption, and then a reintroduction.
Okay,
so the H2 looks like 47-ish here. Yes, Figure 1. This must be open access by now, I would guess.
Yes, it is.
So you can really great figures with nice pictures of humans and pigs and birds, and showing the viruses going between them. All right. So the 1918 virus. They think 7 out of the 8 RNA segments. There are 8 RNA segments in the influenza A virus. 7 out of the 8 probably were of avian origin.
So let's make sure that the Calebs out there have this in their heads.
I can't.
So this is
as a Caleb.
Negative strand virus with a segmented genome, and there's 8 segments, and each one encodes a different gene. And the 2 that are on the surface that are important because that's what you make antibodies to are the hemagglutinin and the neuraminidase, abbreviated H and N, and they're of different types. If we say H1, if you make antibodies to H1, those antibodies won't necessarily react strongly with hemagglutinin that's H3. Or something like that, and the same with the neuraminidase. And all of these viruses, historically, way back, originate in birds.
Yep.
And then they spill over into humans on occasion. And because it's a segmented virus, if you have 2 different segmented viruses that infect the same creature, it's very, very easy for them to reassort segments. Okay, so you can have a virus that's in humans and that human gets infected by a different virus from birds. And since there's 8 segments, it's 2 to the 8th combinations. They can spit out 256 different combinations of viruses, some of which might be successful and present a new problem in terms of immunology or pathogenesis.
Now,
nicely done.
Right, among the reasons they suspect this origin of the 1918 has to do with the U content of the RNA. Influenza viruses that infect and circulate in birds tend to have a lower uracil content than viruses that infect humans. And so when a bird influenza virus goes from a bird to a human, in the humans, the U content, it slowly goes up as it circulates. So you could tell in 1918, these 7 of 8 segments have an avian-like uracil content. All right. And there's one segment encoding for the hemagglutinin that has a higher than expected uracil content. It doesn't match all the other 7 segments. And they think that is because that segment was part of a virus that went into humans in 1905 or earlier, giving it more time to reach the higher U content. That's one of the reasons, and some of the others we'll get to as a reason. Okay? So 1918 virus, 7 segments immediately from a bird, and 1 from a bird, but earlier in time.
So the H1, the hemagglutinin of this 1918 pandemic strain had been around since, what you're saying, 1905 in some other configuration of virus.
That's right. Well,
and then a bird virus, as it were, swoops in.
Very good.
And there's a reassortant event that generates what is going to be the 1918 strain, and the timing of that is around 1915 or so, 1915-16, right?
That's right.
A couple of years before it got really bad. And now we have a virus with the pre-existing H1 and 7 new segments directly from a bird.
That's right. That's right. That pre-existing H1 had been circulating in people. So that's why I had a higher.
And that's, I forget what the previous story was, but this is, there was uncertainty about this before. Lots of different ideas about where this virus came from. This is a new idea, right?
Yeah, there was one idea that all 8 segments came immediately from a bird virus.
I see.
But then when they looked at the U, they realized that probably wasn't correct. And then, as we'll see right now, the seroarchaeology data also confirms this. So again, this is where you take archived sera from people of all different ages. And you can go back pretty far and get sera from people who lived a long time ago.
Absolutely.
And they say that these data have been overlooked. They say that if you look at these data, it clearly shows that H1 of this 1918 virus came into humans earlier than 1918. And in fact, they think it was introduced between 1896 and 1907. And again, this is from archived sera. That's
amazing. In this figure 1, they've got sera that looks like it goes back to the early 1860s.
Yeah.
Yes.
Yeah. I'm amazed that this exists, right? We didn't have freezers. I guess it had been freeze-dried, right?
Freeze-dried, that's right. Just dried.
So they can see a peak of antibodies to seasonal H1 viruses, not the pandemic, in people born around 1904. So it can't be that this H1 arrived in 1918 or shortly beforehand, because we see people with antibodies.
With low mortality, though.
Well, that's a separate issue. We'll get to that.
Right.
We'll get to that. Mm-hmm. Now, other things that were going on that are relevant between 1889 and 1893, despite your year of birth, right?
Yeah, just about right.
There was an outbreak caused by an H3 virus, okay? And then that virus disappeared shortly after 1900. And previously it was thought that this virus circulated until 1918, then was replaced by the pandemic strain, but they say no, that's not true. It disappeared. Again, the seroarchaeology tells us it disappeared shortly after 1900. Now, this is important. The H3N2 emerged in 1968, okay, as a new pandemic virus replacing the H2N2 strain. And if you look at the mortality patterns there, they help confirm this idea that it was an H3 virus that circulated then. The mortality patterns in 1968 to 1970 in people infected with 1968 H3N2, people born after 1900 had no protection, but people born before 1900 were protected in that 1968 outbreak. Of course, there would be people still living, right?
Oh, sure.
They're in their 70s. And if they were infected in, 89 to 93 with this H3 virus, and they were protected when the H3 came back. So isn't it beautiful what you can do with this seroarchaeology?
You bet.
People born in and around 1889, they have antibody titers against H3N8. They think this was an H3N8 virus that was circulating then. It was a pandemic back in 1889. And again, probably didn't circulate up to 1918. And another reason why they think so is that only half of those people born in 1893 have antibodies to H3. If it circulated until 1918, everybody should have it. So they think it disappeared around 1900, this H3 virus. Are you okay so far, Dickson?
Absolutely.
I know it's a little confusing.
No. I know how to count. I still use all of my fingers, and I take off my shoes sometimes, but I'm good on this.
Now, a kind of a side thing here is kind of interesting. If you look at antibodies in these old sera, antibodies to the N8. Remember, the H3N8 circulated from 1889 to 1893. You can find them in people beyond 1900. So they think that an H1N8 virus circulated from 1900 to 1918, and it's that H1 that went into the 1918 H1N1 virus. So that was circulating in people for 18 years. That's why the U content rises. It originally was an avian virus. Okay so far?
Yep.
So basically, there was a single event around 1915 that brought together these 7 avian segments with this H1 from the H1N8 virus that was circulating from 1900 to 1918. 7 avian segments, which they think was from an H7N1 virus that was in birds at the time. And they can tell this by, we obviously don't have any virus isolates from back then, but we can look at what's around now and use the molecular clock, the host-specific molecular clock, to go back and see what was going on. Now, let's a couple of other things that are interesting. They looked very carefully at every RNA segment that we have of both human and swine viruses, and they conclude for sure that swine H1N1, which continues to this day, it started in 1918. It was the 1918 virus that went into pigs. And that's the first time that pigs raised for food were infected with influenza virus, and it was that virus from humans that went into them.
Only humans increase the U content of the RNA rather than pigs? Because pigs and humans are not too far apart, to be honest.
Well, you may not be far apart.
No, histologists have a difficult time telling those.
I think it's mammals. I think it's mammals.
Yeah, I don't think they addressed that in the paper, but it would make sense that it would happen the same in humans and pigs.
As you know, birds are not mammals.
No, they're
birds.
Not. Right. They're distant relatives of dinosaurs.
They are.
Rich knows that, actually.
They're not all that distant.
If you look at them,
actually,
I saw their heads just like a T-Rex.
Alan, I saw a secretary bird on my vacation that looked exactly like a dinosaur.
Big? Is it big?
It's a huge bird. It's about 3 feet tall, and it captured a reptile of some sort and ate it right in front of us. Wow. It was amazing. It was quite an amazing thing.
All right, so the pig virus is, and today they call this classical swine influenza virus, H1N1.
Yep.
It's still around. There are lots of other kinds of flu viruses in pigs, but this one is still around.
Why would it go away?
Well, this one has been around since 1918. Yeah. It went from people to pigs in 1918. And the interesting thing is it evolves very slowly in pigs. And just as a side note, in 2009, when a new H1N1 emerged and caused a pandemic, that H came from this very slowly evolving H1 in pigs, went back into people because our H1 had been evolving faster, and those antibodies no longer protected against that pig H1. So vintages are important in wine and in influenza virus.
Wine and swine.
I had a professor at Mount Sinai, Jerry Schulman. He used to say, "What vintage is that influenza virus?" It was a nice way to put it. All right, so that virus came from pigs. Now, after the 1918 outbreak, it was over about 1920. H1N1 continued to circulate until 1947, and the H1 of that virus did not come from the 1918 virus. It was a very different virus. It came from another virus, not by drift from the pandemic strain. And that's why antibodies to the 1918 virus disappear in people born after 1922 because they were infected with a different H1. So all the 7 segments are the same as the pandemic, but the hemagglutinin segment came from a different virus. I'm looking at you because you're the only person I can
Vincent,
look at.
I'm wide-eyed with anticipation at your next fact.
Now we're going to talk about the mortality. What we learned about this is now where we put all this together and we start to explain why we have this unusual.
Why was this so deadly?
Right. For a particular segment of the population.
The kind of pattern that I mentioned earlier.
Yeah.
Okay. Old people. In 1918, old people were probably protected because they were infected with an H1N1-like virus that circulated between 1830 and 1833.
Look at that.
And they know this from the Sarah archaeology data. People born before 1834 were best protected. It's a similar pattern that we saw in 2009 where people who got infected who were born before 1957 were protected because that H1 was similar to H1 viruses that were circulating before 1957. Now, I happen to have been born in 1953, and so I would be in that category of people, and I didn't get pandemic influenza in 2009. Okay, so I probably, like 1953, I probably experienced an H2 actually, first of all.
You were the epicenter for the beginning of the polio epidemic instead.
The hypernode. All right. Between 1889 and 1893, as we said before, most people born worldwide were infected with an H3N8 virus. If you're born before that, you would be exposed to an H1 virus. Okay? So before 1889, H1. After 1889, H3. This is going to be important in our age distribution. All right. 0% of newborns, 50% of 7-year-olds, and 100% of 14-year-olds had been previously exposed to seasonal viruses in 1847, 1889, 1900, and 1900. And this is going to lead now to their model. So between 1830 and 1847, we have an H1N1 circulating. So if you're born in those years, you're likely to be infected with that. 1847 to 1889, you have an H1N8. 89 to 1900 are H3N8. 1900 to 1918, H1N8. And this determines the exposure patterns of the different age groups that we're concerned with in 1918. So the 1830 cohort, they're infected with H1N1. They're 88 years old in 1918. They would have the best protection. In fact, that's what we saw in the mortality patterns. The really old people who usually get really sick were pretty much okay.
And in 1918, if you lived to be to your 80s, you were old.
That's true.
Yeah, that was pretty good.
You're right.
Yeah.
I think the average age was, 40-ish?
No,
I think they...
Depends on where you're born.
Well, but I think it was still in the 40s, the average lifespan at that point.
1847 cohort, they were 71-year-old in 1918. They were infected with H1N8. They had okay protection because of the H1, but the neuraminidase wasn't matched, so they wouldn't be as protected as the older people. 1889 cohort, these are 29-year-olds. They're in trouble because in 1889, it was an H3N8 that they first got infected with, and in 1918, along comes H1 and N1, and that would make them very sick because they have no immunity. 1900 birth cohort. They were 18 at the time of the 1918, and they got infected with H1N8. They have kind of an intermediate protection because they got H1, but N8 doesn't match up. And finally, infants. They lack protection because they have no prior to exposure to any influenza viruses, right?
But they have maternal antibodies.
I don't
Maybe.
think it helps.
I think maternal antibodies play a big role in flu. Do they?
No, I think not.
I don't know.
I think not because you need respiratory, secretory antibody.
All right.
So they drew a curve, which is in their paper, and it shows the expected protection based on all these assumptions, and it matches the patterns that we see in 1918 really well. All right? And the peak in mortality among the young adults is exactly in those born from 1889 to 1893, which they would be infected by the H3N8, and they're screwed, basically, because that's so different.
Right, because they would have, unlike the kids who are getting the H1N1 for the first time, they would have had a previous exposure to a flu virus, but it's the wrong one.
It's the wrong one.
And so they're going to mount an immune response that's going to be suboptimal because of original antigenic sin, probably.
Yeah.
So no one event explains this pattern. It's an exposure to different viruses over time. And this is all new because we're putting all this data together. We didn't really know this before. Right. So that's the cool thing about this story. So now why does this childhood exposure shape the mortality patterns? As Alan just said, one idea is original antigenic sin. It has nothing to do with religion.
No,
right.
Or Catholicism.
It's a term that's applied to this phenomenon where once your immune system has seen a particular antigen, other antigens that come along, obviously if you get infected with the same thing, that's fine because you've got antibodies that are very specific to that. But if you get infected with something that is closely related, it can be a problem because your immune system will respond with the kind of, sort of okay antibodies that it already has and try to tune those to this different infection. And it would actually be better if it started over from scratch and gave you brand new antibodies.
So if you get, let's say you're a young adult in 1918, you were infected first in 1889 with H3N8 virus. Now, pandemic H1N1 comes along. You make a nice memory response to H3, but that doesn't help you because the new virus is in H1 and there's no cross protection.
Right. So
this is original antigenic sin. That's one idea. The other idea, which they can't rule out, is that now these 20 to 40-year-olds presumably were also exposed to an H1 virus between 1900 and 1918, but it didn't help them because they would still make an anamnestic response to that original sin, the H3 virus. Right. And they say some people may never have been exposed only been exposed to H3 viruses, and the attack rate they say is low enough so that maybe they never saw an H1 virus from 1900 to 1918. So this, I think, is a very nice explanation of where the virus came from and why we have this interesting mortality pattern. And here, we don't have any of these viruses in our hands. That's the cool part. We have sera, and we have sequences of contemporary viruses that we can backdate. And that's how we get all this.
Well, we do have the 1918 virus,
that's the one we do.
That was resurrected. Okay, so I have a question about this because when the 1918 virus was resurrected, the assumption, I think, was that given the epidemiology, that this would be a markedly more pathogenic influenza than other strains around. And a lot of different experiments were done in vitro and in mice, et cetera, to investigate this. And my recollection is that some of those experiments, at least, did indicate that the 1918 flu was more pathogenic than other influenzas around. I don't know that this paper really addresses that. This paper is more about the unusual age grouping of the pattern or mortality rather than the absolute pathogenicity of this strain. Am I thinking about this right?
They do address it, and what they say is you can't really simply put a virus into a naive animal and expect to learn about its pathogenicity in people because their previous exposures are going to modulate the pathogenicity. And in fact, they say that you have to infect an animal with other viruses first and then challenge them with 1918 to really reproduce. And so they say that, animal models are probably not going to tell you because in the end, it's not really the intrinsic virulence of this virus that's making a difference. It is what you were first infected with, and that's the difference. And we've given examples of that throughout. And they have just one more point I want to bring up here. They say this even extends to contemporary strains. So, for example, we have sporadic infections with H5N1 and H7N9 from birds into people. These viruses have never circulated in people, but you can still see interesting patterns. Nearly all the deaths from H5N1 are in younger patients exposed to H3N2 virus, which is a group 2 hemagglutinin. You can put the hemagglutinins of flu viruses into two groups, group 1 and group 2, by their sequence patterns. H5N1 is a group 1 virus. H3N2 is a group 2. And if you get infected with the group 1, you're less likely to get really sick after an H5N1 than if you're infected with H3N2. So that's a similar idea. Same thing with H7N9. It's a group 2 HA. The mortality is mostly in people infected with a group 1, which is either H1N1 or H2N2. So I think these are really significant, not just in explaining the mortality and saying where the virus came from in 1918, but the idea is maybe we can modify immunization in such a way so that that first experience, if that is your first experience with flu, your flu vaccine, you can make it so that you will subsequently, that response which you'll get on challenge from another virus in the wild will give you the right antibody response to neutralize the strains. So that's really, I think, the importance here is that we can learn to make a vaccine that better suits this pattern.
Right.
One thing I wanted to talk about, and I kind of went off on a goose chase about this, is I learned this fact that the uracil content tends to increase after avian segments are transmitted to mammals. And so then I was tracking down the paper that describes that. And because I kind of wanted to know more about why, and essentially in the end, they just give three possibilities. One that they don't think is due to a structural thing that that with more U's and fewer C's, that could impact the RNA structure, and that might explain that difference. Another is that the RNA polymerase machinery has different cellular components in human and avian cells, creating a relative mutation bias. And then another speculative possibility is that humans have a native defense against RNA viruses that operates similarly to the ApoBec family of genes, which causes deamination of cytosine, resulting in uracil. So I didn't find an answer, but I did think that fact was interesting, and they use that as corroborating evidence for some of their clock-type data, as I understood it.
If I'm not mistaken, that was done by Arnie Levine and Raul Rabadán. Is that one of the papers you found?
Rabadán is the author. Yeah, Rabadán, Arnie Levine, and Harlan Robbins. Yes, you got it.
So Raul Rabadán is here at Columbia. He was on TWiV many years ago. Many, many years ago before No. No, not that.
So one more thing for any listeners out there. I found it hard to get all this together. This is open access, and there is a summary figure 3, that really does a good job.
Yep.
Yes.
Of summarizing the whole thing. So if you're really interested and you feel like you've not quite got it, have a look at that paper and look at figure 3, and it really paints a pretty clear picture. And
spend some time parsing figure 3, because there are multiple dimensions of data on it that, it's not, as we've said before about figures and papers, these are not magazine illustrations. These are data. So you're not expected to glance at that and get the whole meaning in 10 seconds. You take 20 minutes and go through that, and it really does do a really nice job summarizing.
But we won't mind if you just listen to this TWiV episode.
And that's fine,
too. That's good, too.
Because if you're driving, that's probably all that you can do.
Right. Don't go looking at the paperwork.
And if you are listening, you're about to hear me read an email.
Go ahead, Dickson.
So Waitama writes, "Hi, y'all." It's not how you'd say that from Australia, but that's okay. "I'm writing this to you on the plane to Hungary. The weather on arrival should be clear skies and 28°C. 2 other PhD students and I from James Cook University, Australia, are on our way to attend back-to-back conferences in Budapest. The International Symposium on Viruses of Lower Vertebrates and the International Symposium on Ranavirus, I presume that's frogs, are running from the 4th to the 10th of July. Can you please give a shout-out to all the veterinary and wildlife virologists in Budapest this week? And remember, it's not only men and mice that get viruses. Cheers from a jet-lagged student, Watama."
Well, shout out.
All right.
Shout now.
But 4th of July is a ways away.
Yes.
It is.
And I guess you're going to have a little fun there in Budapest. But they have good food there. Right?
Wonderful food.
Let me see. Have you been to Budapest?
I have not, but my wife's daughter spent 6 months there as an exchange student, and they visited her twice, and they said it was just magic.
I bet Kathy's been there.
I have been there.
See?
We had an adenovirus meeting just outside of Budapest. It's really great.
Yeah.
Now, are viruses of lower vertebrates responsible for back pain?
Oof. Oof.
Did you just think of that?
That's
I did.
pretty good, actually.
That's very good. I like it.
Yeah.
That's vertebrae.
Oh, the right. Yes. Okay.
Alan, could you take the next email, please?
Sure. Audra writes, Dear TWiV, I'm a longtime listener of TWiV and now TWIM. I just read this article in Science, and I'm absolutely heartbroken. And this was, this is about the, this is about the situation in Nigeria. It's a famine going on there, right?
Yep.
Yeah, basically it's famine and the interplay between famine and disease.
Yes.
And the cause of the famine being the political unrest in Boko Haram.
It always is.
Yeah.
Yes, okay.
5 million
So
people
there's
are starving.
Yeah.
Yep. And there's
and that's the richest country in Africa, by the way, because of the oil. Just
they
remember
talk
that
very
too.
specifically about malaria,
Yep.
measles,
Yep.
and polio.
Yep.
And Audra continues, there are so many infectious diseases rampant in Nigeria, malaria, measles, polio. Is there any way the TWiV listener community could help out? I don't really have a solidified suggestion or an idea. I'm writing to you in a moment of sadness and wondering if there's any way, as virologists, microbiologists, we could all help out this humanitarian crisis. It's one of so many right now in the world.
Yeah.
And the really tragic part about this is modern famines. I don't think that in the past century we've had a famine that was actually due to a shortage of food.
Right.
That's not shipping food to Nigeria will accomplish nothing except wasting food, because it won't get to the people who are starving. The reason these people are not getting food is because their government is not working for them.
Right.
And as Dickson said, this is the richest country in Africa. Is it richer than South Africa?
Yes.
It is.
Okay. So, and it's from oil money, which makes a tiny number of people extremely wealthy and corrupt in this case, and everybody else is pretty much left to fend for themselves, and millions of them are now starving, and that is not going to get fixed until somebody deals with the corruption at the top, and I don't have a solution for this either.
No.
This is something that requires international action, which is sadly the governments of the world and the one that would normally take the lead on something like this are not in a position to do anything right now. So there we are. But yes, the disease aspect of this is compounding the tragedy.
All right, the next one's from Anthony, who sends an historical review of polio from The Independent. Independent.co.uk. And a quote from it, a passage in Philip Roth's novel Nemesis describes the horror of catching polio in Newark, which is, of course, New Jersey, in 1944, when outbreaks of the disease were common and each summer was spent in fear of infection. Is there a Newark, Delaware also?
Newark.
Yes.
It's not Newark. It's Newark.
In Delaware, it's Newark?
Newark.
And here in New Jersey,
do not
it's Newark.
want to be confused with Newark.
I like to be corrected. It's fine.
No, it's spelled the same way. It's just pronounced differently.
There you go. Famous polio survivors: Francis Ford Coppola, David Starkey, and Mary Berry. I didn't know that. And Anthony also tells us that Linus Pauling is the only person to have been awarded 2 unshared Nobel Prizes, 54 in chemistry and 62 peace. And that is in response to something we said a long time ago, which I don't remember. But thank you. Kathy, can you take the next one?
Yes, Gary writes, hello, Twivers. I was listening to an old episode today and heard a letter talking about Amazon. Is that how you pronounce it? I don't know. Amazon, and how you could not find information about it. I'm talking about episode 59 and an outbreak of swine flu in the Ukraine. I decided to see if I could find any information about the medication, and I found it. I'm curious what you guys think about this drug. Here's the link. I hope you'll let us know what you think.
It's snake oil.
And someone has typed in snake oil.
That's right. That was me.
The reason I wasn't sure of how to pronounce it is capital Amizon. So at first I thought it was a typo in the person's letter, but that's really how it's spelled.
Right.
So it is, the active ingredient is N-nisaminum.
N-nisamium iodide.
N-nisamium iodide. Fairly simple compound. This has been used in the former Soviet Union and marketed there as well. And it is said that they have clinical trials that attest to its efficacy, but I could not find any. Well, the very few things that I could find were a bunch of in vitro studies that say that it's ineffective against flu. The link that Gary sent is from a site called Medicine Worlds that looks like it's
I think it's a Russian online pharmacy.
That's right, a Russian online pharmacy. And if you look down further on that site, one of the giveaways here is that it cures everything.
Mm-hmm. That's a sign.
Medicine hack from Russia.
Thank you, Gary, at least,
hack.
but thank you for looking it up. We appreciate that. 59 is a long time ago.
It is.
Rich, can you take the next one?
Hang on. I have are you there with me?
We're here.
We're here. Where are you?
I just got a warning saying that I've been infected with a virus.
Oh.
From this Amazon site.
Just close it.
Kidding.
Yeah, I'm going to.
Is it a double-stranded RNA virus or is it a single?
It's a rhinovirus.
It's a runaway virus.
Actually, it's frozen up my
All right. So you want to reboot?
my browser.
Just quit your browser. Force quit your browser.
I'll read the next one.
Read
Mary Claire writes, "Hello, Vincent and colleagues. I'm writing to thank you for making such worthy podcasts. I subscribe to all the TWiV shows, and I've come to expect at least one personal light bulb moment every week while listening. The papers are always interesting, and the conversations between the hosts are good fun. I've learned an enormous amount from your podcast, and it's time I thanked you for the exceptional work. Cheers to the whole team. Mary Claire Rollins, Montana State University." Let's hear it.
All right.
Thank you.
Alan.
Stephanie writes, "Hello, I'm not a scientist, but I have the flu and stumbled on your March 31st podcast." I'm very sorry to hear that. The flu part, it's fine, you're not a scientist.
Exactly.
I was wondering, why am I so sore? I literally have physical pain up and down my spine like I've been hit by a car. Please help me understand how a virus can cause physical pain.
That's a lower back pain virus there, Alan.
Yes, exactly. It's a lower vertebrae virus.
There you go.
It affects your lower vertebrae.
Lower vertebrae virus.
No, I think that because you make
achy.
cytokines when you're infected, and these diffuse from the site of infection, and they have global effects.
They do.
Including causing muscle pain, which is probably what you're feeling.
Yeah.
Would anyone like to elaborate, or does that make sense?
That makes sense.
Yeah.
Makes sense.
What do you
to
think,
me.
Kathy? Yeah.
Yeah, inflammation, edema, all those kinds of things. But it's funny because what I've always told people in my classes about influenza signs or symptoms is that you feel like you've been hit by a train, and she felt like she's hit by a car. Yeah. And also supposedly that you can really pinpoint it, almost to the hour, is when that happens.
It's true.
Rich has just pasted in a little information.
I pasted that.
You pasted on how to force quit.
How to force quit an application.
Rich is still working on it. So in the meantime, I will take Harlan's. Dear TWiV, thank you kindly for your informative and engaging discussions of virology. As an undergraduate in clinical laboratory science, I'm eager for the sorts of discussions you provide across the whole TWiV spectrum. Seasoned scholars discussing the nuances as you do helps inspire me by elucidating the world that I'm steadily exploring with fun yet deep details of microbial machinery. Keeping with tradition of mentioning local atmospherics, it's 26C here in Honolulu. Also, keeping with tradition, I believe that Vincent should also be nicer to Dickson, whose commentary in particular I treasure.
Hear, hear.
The political commentary is slightly longer than I prefer, if only because the intersection of science and politics at this time is somewhat despair-provoking. I agree with the principles that you've mentioned across various episodes, though. Having been so inspired by TWiV, I've just purchased a copy of Principles of Virology that I've had my eye on for several months. Should any of you ever grace our tiny island, I'll happily take whoever makes it for beers.
Consider that done. That's being nice to Dickson.
And Harlan is from Iwa Beach, Hawaii.
I'll read the next one from Laura. Doctors Vincent, Racaniello, and Dickson Despommier. I recently found TWiM, TWiP, and TWiV while adding podcasts on aquaculture to my list. I finished my laboratory sciences degree back in 2010, and I'm quite sad I hadn't come across you sooner. I find myself addicted to your shows, to now include urban agriculture. Despite my addiction and subsequent strife of not having enough time in the day to podcast and binge, I would like to extend my gratitude. I delight in the histories and stories previously unknown to me, as well as your humorous and informative banter. I read these articles last week and thought of you too. Something about Theileria parva, yellow fever, rat lungworm, and measles.
It covered the gambit.
Yeah. Thanks for all that you do, Laura.
But Laura, there's Rich and Alan and Kathy also here.
There is.
But we're not on TWiP.
But you're on
right.
TWiP.
Oh, but
you're on
still.
TWiP. She listens to TWiP.
Yeah.
All right, Rich, the last letter. You can read that. We'll be done with the letters.
Right.
Okay. Ryan writes, Dear TWiV hopes, I hope this is gender neutral enough. If not, I will use TWiV hostess. I have several comments to the past few TWiVs. 1. TWiV 437. WWTP is a common abbreviation for wastewater treatment plant. I am currently working as a postdoc in the Water Desalination and Reuse Center in Kaust.
Reuse Center.
Reuse Center in Kaust.
They say they're desalinating the water.
Yeah, that's right, but they're just reusing it.
It's a ruse.
The Kaust is the King Abdullah University of Science and Technology in Saudi Arabia. Nice. One of my projects involves studying the different viruses present in a wastewater treatment plant. We studied the different viral species entering the wastewater treatment plant in human waste, the viruses which are retained in different stations of the wastewater treatment plant, the viruses which are exiting the wastewater treatment plant, and the viruses which are inactivated by the wastewater treatment plant. I'm sure by now you must be sick of saying wastewater treatment plant and appreciate This is extremely useful in writing manuscripts on WWTP efficiency with really low word count limits. 2. TWiV 435. You brought up the plaque video, which was discussed in TWiV 68, Ode to a Plaque. I remember hearing this TWiV as a miserable PhD student and showing it to my supervisor. He then used it in a lab practical to teach about plaque formation.
Nice.
Fast forward several years, I was invited to lecture on Introduction to Virology in a public health course in my current institute. I used the same video for my lecture, and I still get memorized by it. In fact, after showing the video, I played it again just to observe the awesomeness of plaque formation.
Right.
I think you have many people doing the same thing. I used that movie.
One more small correction, Rich. You said memorized. You meant mesmerized.
Mesmerized, sorry.
Be nice.
Nice. I was very polite, I thought.
That's cool. 3, I can't remember which TWiV where this was discussed, but the topic of digital PCR was brought up. One of the hosts mentioned that the machine is expensive and has to be in a core facility to be shared by different groups because of the cost. I'd like to say that we have a DCPR machine in our lab, not sharing with anybody else. This DCPCR machine, DPCR machine, runs on a different principle than the one discussed on TWiV, which is droplet-based. Our machine runs by partitioning the DNA template on a chip. He gives a link to it. This machine costs approximately 5 times less than the droplet-based digital PCR machine. Still rather expensive, but funding is not a big limitation to research in Saudi Arabia.
Right.
Maybe the United States should follow the same model for research funding as Saudi Arabia. Tell that to the far right. 4. Lastly, I'd like to express my appreciation to Professor Rich Condit. Nowadays, it seems like if one wants to be successful in research, one should sacrifice family time. But on several occasions, Professor Condit had mentioned how he made sure that he allocates time for work and family, even as a young researcher. I specifically remember Professor Condit explaining that he was in charge of baths before bedtime when his children were younger. This meant that he had to return home early and make the time at work really productive. I totally can relate to this, as I am in charge of bedtime readings and school lunch preparation. I do not want to push all this to my poor suffering wife, who also has her own career. I also follow other advice which Professor Condit has given, like put away frivolous matters at work and focus on research, and to come in for a few hours over the weekend to catch up on any work. I feel that young parents should really embrace and enjoy the time with children when they are young, because this time will never come back, and I look to Professor Condit as an inspiration for this. I also hope that in retirement I can spend time traveling and visiting my children and grandchildren just like Professor Condit. One can spend time with family and still do well in research. The pressures of publishing can get to a young researcher, but spending time with a family, I find, is the best way to beat the stress. Sorry for the long emails. Regards, nice. A huge fan, Raihan. Sadly, I'm not working on influenza B viruses anymore, but they're still my babies. Oh, he was the capital B letter.
Yes,
right.
That's right.
He won a copy of viruses. Hey, Ryan, did you get it? Just tell us if you got it.
Exactly.
Never heard. There's another advice of Rich Condit came up recently. I was interviewing people for the NITO TWiV last week, and I asked them, how they got into a lab, and Margot Brinton said, I started by washing glassware. And I said, that's what Rich Condit tells people to do.
Wash dishes.
Isn't that cool?
That's right.
I actually started in a lab washing glassware too, and I had never connected that with Rich telling people to do that.
There you go.
But figure this out. I never learned how to use a lab dishwasher. I had to wash the dishes by hand because
heck yes.
I was a physiology lab, and where they were concerned about the sodium and potassium and everything. But throughout my career, I have never learned how to use a lab dishwasher, which means when there's a problem, I can't do it.
My first job in a lab, I learned how to wash cover slips to microscopes.
Did you break a lot of them?
Yeah, I did.
Why do you have to wash them?
Said, you don't do this job anymore. You can wash the slides, but not the cover slips. But I thought that was actually inviting contamination for the next diagnostic go-round. So
you broke them?
No, eventually we ended up not doing that. But the person in charge of the lab was exceptionally frugal and thought that washing cover slips was a good idea.
You wash tips also?
We didn't have that thing in those days. No, we just had pipettes. We had glass pipettes.
Yeah, I know. I was in that era.
We used to soak them and reuse them because the budget was very, very low. This was a hospital budget we were dealing with, so we couldn't be expensive in our thinking process.
We had these reusable glass pipettes. I forget what they were called, but you would soak them in Chromerge, right?
That's
exactly right.
Chromerge, yep.
Got it.
And then
automatic pipette washer would go.
Yeah, you had this thing where you could stick about a dozen of these things in the pipette washer and go from bucket to bucket.
By the way, I know the person who invented the automatic pipette washer. Because his job was to wash the pipettes. It was Elmer Pfefferkorn up at Dartmouth. He invented the automatic.
Was it Elmer, really?
He did.
Wow.
He did.
Someone's going to get mad that we're geeking out here.
No, we're not geeking
about pipette
out.
washing.
So nowadays, no one washes pipettes because plastics are cheaper and
disposable.
Right.
Yeah. Right.
We still do in my lab. Yep. The Holland, John Holland wanted to pay undergraduate dishwashers rather than pay the oil companies for the plastic. That was his philosophy.
So the pipette washers are cool because they're a big plastic thing. You put them in,
love it.
And then it fills and then automatically drains. And I remember walking past
that's good stuff.
watching these filling.
That was good stuff.
Because you had to rinse them many times to get the Chromerge off.
We actually had glass pipettes still in the lab. When I was there, and we only used the plastic ones under the tissue culture hood.
Yeah, For macro pipettes, I used glass. Even in the tissue culture hood, I used shorty pipettes
Shorties,
all
yep.
the way.
Shorty
My whole career.
Yeah, Alan, before you got here, we had these shorties for the tissue culture hood because the long ones are kind of awkward in the hood.
Yeah.
Yeah.
They are.
Yeah.
Do you have shorties, Kathy?
Yep.
I got lots sitting around. You want them? Nope.
No, we probably have enough.
All right.
But if we were thinking about buying any, I'll let you know.
I mean, no, I got
They rewash them.
big and little
when they
ones.
recycled their.
Man, I had some shorties that were so used you could barely read the numbers on them anymore.
Yeah.
You just kind of know where 4 mLs was.
Yeah, right.
All right, let's do some picks. Alan, what do you have for us?
I have what I guess was probably one of the very first computer games of any kind. Colossal Cave Adventure is now open source. Now, a lot of listeners probably don't remember this or weren't even born then, but this thing was developed in 1976, and I remember playing it in the 80s in college on a mainframe computer. This was a text adventure game, and it gave you a description of where you were. You know, you're standing at the end of a road before a small brick building is how the thing starts out, and you give it 2-word commands, and you walk around and you explore this cave. It was actually based on one of the programmers was a caver, was into spelunking, and he came up with this whole. He was also into Dungeons and Dragons, I think. So he came up with this whole adventure game that was based on using the things that you find to solve various puzzles, and it's all text. But a very important piece of computer history and game history. And finally, some folks got it converted to open source and uploaded it to an open source repository online, so people can now get the source code for one of the very first computer games, and I think that's pretty cool.
Look, they're running it on a VT100.
Yes, that's the way it should be.
This looks like what evolved to be Zork.
Yes.
It reads like
Infocom,
Zork.
Infocom grew out of, and also the Scott Adams Adventures, not Scott Adams of Dilbert, but there was a different Scott Adams who did adventure games on the TRS-80 and Apple II Plus platforms. And Zork, of course, was also on those, and Infocom was the company that did Zork and then did a whole series of additional adventures.
Did this predate Dungeons and Dragons?
No, this was, I think Dungeons and Dragons was early 70s, right?
Yeah, it was the same kind of game, right? Text-based.
Right. Yeah. I think the introduction of Dungeons and Dragons, 1974.
This photo is old because you can see the tapes on the shelf behind the channel. I love the last part. You scored 27 out of 350. You were obviously a rank amateur. That's great. I love it. So you can play this on Mac OS, Windows?
You can play this. Well, you could download the code and modify it if you wanted to, and you could port it over to whatever else you wanted to play it on. But don't. Yeah. Actually, this one I guess you could play in a completely dark room. Don't play Zork in a dark room because you may be eaten by a grue.
Yeah.
You hear that, Dickson?
I did. I will make sure I don't.
Rich, what do you have?
Apropos of our interview with Kate Rubens 2 weeks ago now, I have a Time Magazine feature called Life on Mars. It's one of their 360 features where they have videos that you can pan around in. There's going to be 4 episodes in this. They've published 3 of them so far, and it's about a project that's funded by NASA that's one of these analog projects that is supposed to simulate life on Mars, where they have something like a habitat that's on the Mauna Loa volcano in Hawaii.
Yeah,
so I think the University of Hawaii has probably obtained funding from NASA to do this, so it's a collaborative. And I've got a link in here not only to the Times article, but to the Wikipedia article on this project called High Seas, which is the Hawaiian thing. What is that? It stands for Hawaiian Space Exploration, Analog, and Simulation. They are now up to High Seas 5, and that's what this thing is about. So they've had 5 different adventures with this thing, where they take a bunch of people and they put them in this habitat, and you have to stay there, and you can go outside, but you have to wear something akin to a spacesuit. Right.
That's true.
And the longest of these so far was a year.
Right.
They have a couple of 8-month guys. The one that's going on now is 8 months. There are 6 people involved. And like the other analog mission that I picked a few weeks ago, the idea is to find out how people react to being in a habitat and a confined environment with other people for a long period of time.
Not well.
Should have asked Nelson Mandela.
It's interesting. When we asked Kate, would you go to Mars?
Yeah.
Her answer was, yeah, with the right crew.
That's right.
Right.
Okay, so that's what it's all about.
And this is the right location for this because that area it looks like a desolate planet.
That's true. Actually, there's one other place that's better than that, to believe it or not, and that's the dry deserts, the dry valleys of Antarctica.
Right.
Which is as cold as Mars.
The advantage to this is you could actually, if something goes drastically wrong, you could get emergency services in there.
Exactly, but not in
Antarctica.
Mars.
No, not Mars.
No, that's why they're doing these experiments.
That's why you test it here.
That's right.
Because they want to set up labs on Mars.
And that's a great follow-up to my pick of the week, because I didn't list it, but I will now.
Go ahead.
There's a wonderful new initiative by probably the country of China, but it's being focused in Shanghai. And they're setting up over 600 acres of training center for urban agriculture with vertical farming as its centerpiece. Mm-hmm. And if you go online and just type out Shanghai vertical farming, you'll see it. And it's absolutely stunning. And I'm just speechless over the complete acceptance of the idea, and then the next iteration of this, they want to be the training center for how to grow food in urban centers.
Shanghai Vertical Farm.
Shanghai Vertical Farms. That's right.
All right. Have you been?
No, it's not built yet. It's been designed. I believe they've had groundbreaking, and they're going to start building it. In the next 5 years, you'll be able to go there and train to grow food in buildings anywhere in the world.
All right. We'll find it.
So the reason why they're doing this is because this Chinese space agency wants to live on Mars, and you can't do that without having these buildings there to help you out. Nobody just eats potatoes.
Shanghai is planning a 100-hectare vertical farm to feed 24 people.
No, that's not it.
24 million people. I'm just kidding.
That's 24 million people.
A massive 100-hectare vertical farm. Is that it?
Yeah.
There are lots of news articles.
Yes.
Is there any particular one that you like?
The one that features the architect that's in charge of the design for the whole thing is the one I've picked out, because they got pictures to go along with it. It's quite remarkable. Yeah. By the way, I should mention the fact that I was on vacation, but I actually gave 2 talks. One was in Milan at a meeting called Seeds and Chips. Well, thing seeds and chips, you think, what the heck is that? It's the largest indoor grower meeting in the world, and you shouldn't laugh too much at this, because on the second day of the meeting, they devoted 4 hours worth of time to Barack Obama, who just sat and talked.
For 4 hours?
Yeah.
Did other people speak with him?
No. Yes, his chef that he took with him from the White House. And they reminisced about the days in the White House and the way the policies were made and why. Politics is politics, he says. They're going to try to change things, the next regime. And he wasn't bad-mouthing this regime at all. As opposed to in Fresh Air yesterday, Biden was interviewed by the host, and he couldn't stop talking about the evil.
Did you listen to all 4 hours?
I had to. I was sitting in the audience. I was right down in the front.
It was probably interesting, right?
It was fabulous. It was absolutely fabulous. And he's such a wonderful speaker.
Your Columbia professor?
No, I didn't get to meet him.
Did he yell? Hey!
You should have seen who was ahead of me in the rows. All of Milan showed up, including the mayor and all these people. So it was really, it was quite amazing. All the Secret Service people were still over the place. Yeah, it was nice.
Kathy, what do you have?
I picked something that I found a couple weeks ago when we had the ladybug discussion. And I always find it interesting when I come to a website and it has what I want, and then the origin of it in this case is a University of Michigan thing. And so that was partly why I picked it. But it's called it's a BioKids website. The link that I put in is to the critter catalog. And if you go to the bottom, you can go to the homepage. But it's a huge repository of things about mammals, birds, reptiles, amphibians, annelids, mollusks, insects, arachnids, and so forth. And it's kind of small print, so maybe that means it's for kids. Because we older people want larger print with, bad eyes. But just got lots of information, and there's a search box so you could put in ladybugs or whatever. And it's, to some extent, I think, focused on specimens, or critters in Michigan, but I'm sure that, I know that many of these are found outside of Michigan, so it's not just going to be a Michigan-specific tool. And it's sponsored by University of Michigan, Museum of Zoology, the University of Michigan School of Education, and Detroit Public Schools. It's a partnership of all of those. And National Science Foundation is contributed some of the work that goes into it. So it's just a pretty cool site with lots of information.
This is really cool.
Common animals in Michigan.
Yeah.
No people, though.
I'll bet you, though, Kathy, that there are no wolverines that still live in Michigan.
Oh, I don't know about that.
Why would you say that, Dickson?
Because they were all shot.
By whom?
Who do you think?
Hunters?
Mostly farmers.
No results for wolverine.
Nope.
There are spiders, though, Dickson. There are
tons of spiders.
Arachnids.
Cannot get rid of them.
And
the
fishers.
moose is the moose is very prominently featured under mammals.
Moose is very big.
A lot of pictures of mooses.
Paradomestic animals. They've become paradomestic.
Is that right?
Yeah, they have.
Name another peridomestic animal.
Deer.
Right.
Thank you, Alan.
Mice?
Rats. Pigeons.
Yeah.
Sparrows.
You wouldn't consider bats peridomestic, right?
No. Well, they do live near people. Some of them.
Yeah.
Some of them. Little brown bats do, but mostly the fruit bats don't.
Cool. All right, my pick is TWiV 154 with Arturo Casadeval, which we recorded at New Orleans. And the reason I picked this is not simply to promote other shows, but we have talked about Arturo's work, his papers before on TWiV, particularly the ones about reforming science. He writes a lot of stuff like that. And we talked mainly in this episode about things he's written on, like moonshot science, the war on cancer, why those kinds of things are good. Rigorous science, call for being more rigorous in science. How do we do it? And he's got some really good ideas about that. Funding by lottery. I think this is a great idea.
Yes.
That would be good.
And finally,
take the top 30% or whatever, and
mix them in a bag and
draw.
Wouldn't that be
names
great?
Out of the hat?
Because basically, it's a lottery now, but it's not a fair lottery.
We used to call it a crapshoot anyway.
And finally, the pathogenic potential of a microbe. So he doesn't like using measures of virulence that we have for bacteria or viruses or fungi, and he proposes this number, which is pretty neat. And it's because a microbe has no virulence without a host. So it's always a pathogenic potential. Kind of neat. So he was there and he talked about it with us. And he's a very interesting fellow. Of course, he's originally from Cuba. And he's never been back. I asked him. He grew up here in New York City. And he said, I probably won't be able to get back ever again. Because, of course, we were reversing the
I don't know.
open borders. Just said today. They're reversing it.
Yeah.
You don't think it's going to happen?
I know. I think you will be able to go back. You will.
Anyway, Arturo, and we had Michelle Swanson and Elio Schechter and Michael Schmidt and myself. It was a lot of fun. We had a nice little audience. Check it out. And we have a listener pick from Grant McFadden. A bit long for a pick in TWiV, but a nice summation of how complex it is to ensure data and reagent reproducibility in science using antibody validation as the exemplar. And this is at BioCompare. It's a video where they talk to people and say, what are the problems with buying antibodies from different sources? How do you make sure they are what you think they are? Which is something we've talked about here. There used to be a website that compared antibodies, right?
It's an enormous problem.
Does anybody remember that?
I am actually working on an article on this very subject right now.
There you go.
There's so much controversy.
And a big part of the issue is it used to be 40, 50 years ago, if you needed an antibody against something, you made it in your own lab.
Right.
Right.
And so the validation, well, you made it. You obviously tested it for the use you wanted.
Yeah.
But there's now, there are so many proteins that people want to work on, and everybody's doing things at such a fast pace. There's a whole industry that sprang up to cater to this.
That's true.
It turns out that Wuhan, China, has become a major center for manufacturing of antibodies. And the business operates very much like consumer products, where you get this thing that's made in one factory that you've never heard of, but then it's sold by some other company that you have heard of. You call up your lab supplier, and they've got all thousands and thousands of antibodies listed. And so you order an antibody from them, but they didn't make it. They bought it from this manufacturer and rebranded it and put their own catalog number on it. And if it doesn't work, then you say, well, okay, I'll order one from another lab supplier. And you order an antibody against the same protein from another supplier. It's got their brand name and their stock number. What you don't realize is that it's exactly the same thing from the same batch from the same factory.
Wow.
It's just being resold by different suppliers. And then on top of that, people tend to take for granted that the stuff they're buying is going to be useful for their particular application, and they don't necessarily understand how to analyze that. So researchers themselves share some of the blame in this. Publishers are partly to blame for not insisting that people document their methods properly. It's quite a big mess.
So you probably know that in NIH grant applications now you're required to fill out a section called how do you know your reagents or what you think they are?
Right.
And if you have antibodies, you have to show, we did a Western blot with purified protein, and it does recognize that protein. And I guess this is in part the reason for that.
Yeah.
So Grant has moved to, I'm sorry, go ahead.
I was just going to say that document that you provide for your grant is discussed after the scoring.
Yes.
In the same way that budgets are. So right now that kind of information is not counting into scoring.
Yeah, it's important though, right, to make sure people have, and then you could say in the critique outside of the scoring, this, you should validate this, give people a little advice.
Yeah.
Now, Grant has moved to Arizona State University in Tempe, Arizona.
It's a wonderful place.
And he sent us a list of the upcoming temperatures in Tempe: Thursday 108, Friday 109, Saturday 110, 114,
That's right.
118, and 119 on Tuesday.
Yeah.
That's not a very temperate climate.
But it's a dry heat.
It's a very dry heat. My son,
but so are ovens.
my older son was in the Air Force, and he was stationed just outside of Tempe. And many times they prevented them from flying because they were afraid that the tires would deform on the tarmac.
It's pretty hot.
It got up to 140 to 150 degrees Fahrenheit on the tarmac.
In hot weather, too, you get less lift.
You get less. There you go.
It's a really serious problem, especially in parts of Arizona that are high altitudes.
So
then you get thin air and hot air, and you end up not being able to take off.
But Saudi Arabia has an Air Force.
Oh, sure, they do.
Works.
And they have to load their planes with this in mind.
Yeah.
And do you use flaps to accommodate for that? Do you extend them for better lift?
Well, it depends on the airplane.
It was Luke Air Force Base.
You actually have to go on a hot day, even at the near sea level airports that I fly out of and the little planes that I fly. When it's hot in the summer, and if I've got a couple of people with me, I'll go to the book and look up the numbers and say, gee, can I use this? Can I use this runway? Can I get off the ground? Because the worst thing is if you start the takeoff thinking you're going to be fine, and then you're running out of runway and you're already doing 40 or 50 knots and you can't get off the ground, you may not be able to stop in the remaining runway. And quite a number of accidents have happened that way.
That's a problem.
It's a very big problem.
It's a very serious problem.
Very big problem.
It's too bad for University of Florida. They lost Grant. It's too bad. They lost Grant. They lost Rich Condit.
Right.
Any poxvirus left there?
No, as a matter of, there's Nassim. He's still there, and there's a segment. Grant's got a project left over there that'll go on for another year or so. But no, that's, in the heyday, it was me and Grant and Dick Moyer, and there have been some other people there as well who sort of played around with pox, but those days are gone. But we've got, Scott Tibbetts and Stephanie Karst and Dave Bloom and Rolf Rennie, and others. So there's still a strong virology community. They need to recruit some more, but it's still a great place to be.
That is TWiV 446. You can find it at Apple Podcasts and microbe.tv/TWiV. Interesting development, by the way, in terms of podcasts. The most popular player for podcasts is Apple's podcast player. And, there are many others you can use to listen to podcasts, both on iOS and Android. But Apple announced they're going to be releasing detailed statistics. So now we know how many downloads there are, but they're going to tell you how long people listened for and when they stopped in the episode. So we'll be able to find out if people actually listen to the picks or the letters.
I'm not sure I want this information.
I know.
Exactly.
The problem is, Alan, that advertisers are going to use it. Now, now they use download numbers. They say, you need this number of downloads to get an ad.
That's one of the reasons I'm not sure I want this information.
And now they're going to say, well, your people only listen to a quarter of the show. We're not interested. Wow, that's going to be tough. So it's coming. Now, it's only from Apple Podcasts, and we're not going to know what goes on with all the others, but so be it. Anyway, send us your questions and comments. TWiV at microbe.tv. Consider supporting us. We'd love to get your support. Notice we don't have a lot of ads these days. We have one a month from Blue Apron, and that's it, because we don't have enough downloads. Your network is too small, so we need your support. Go to microbe.tv/contribute. Give $1 a month. Give $1 a month, say, for the next 40 years, and we're all set. If everybody did that, we'd be set. Okay, Dickson Despommier, he is at verticalfarm.com, parasiteswithoutborders.com, and thelivingriver.org. Thank you, Dickson.
What a pleasure to be back. It's
good to have you.
Had a lot of fun.
I was nice to you.
You were very nice to me. In fact, everybody was very nice to me this time. I think I'll come back to the next one.
Your first week back.
It's kind of like a honeymoon. Is that it?
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit, formerly University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure thing. Always a good time.
And Alan Dove is at alandove.com. He's also on Twitter as Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. The introductory music on TWiV is composed and performed by Ronald Jenkins. You can find more of his work at ronaldjenkins.com. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.